HIV. Uh, so at that point, David Wolski jumped on this. Actually, he was the Epstein-Barr virus guy. he had his postdoc from Karolinska Institute, and then the very promising young virologist. So he is one of the person who developed one of the first antibodies that were used to detect HIV, and the RNA hybridization assays they were used for the infected population at multiple places. So very soon he developed interest in neuropathogenesis with HIV, and particularly his interest was how HIV interacts with the non-neuronal cells like astrocytes, microglia, or perivascular macrophages, and that lead to the neuronal dysfunction and eventually the dementia. But more than that, these days he is more famous for the development of this mouse model, eco-HIV mouse model. And that is also a reason why he was invited for today for this seminar. As I should note this here, that more and more of the investigators who submit the pilot application to DC CFAR are interested in using the eco-HIV mouse model for their studies. And this mouse model is taking a lot of traction in the field. And so we thought that it will be great if we hear from David Wolski about the newer development of this mouse model. So without further ado, I'd like to welcome Dr. Wolski, and here you go. Thank you so much.
Thank you very much for inviting me, and it's really a pleasure and privilege to talk to you, and especially talk about this model, which, as Sanji knows, was developed together with Mary Jane Potash, co-scientists who work together, also are married. And I will have just a little bit history of this in my lecture. and for those of you in obviously virologists, you know how against the grain it was to try to develop a mouse model of HIV based on virus infection of mice. But we persevered, and you will see some dates of those publications. And I know that like every model, it's just a model, right? And does not reflect 100% everything that HIV does, but I think allows us to, especially neuro-HIV field, be very close to mimicking what happens now in people in art. And I think in other, perhaps the model will be more recognized there is a dichotomy between institutes and NIH institutes that fund us, as you can see by the names here at the bottom of the slide, and very difficult to get funding from NIAID. Okay, so classical retrovirology people still don't like us, but I have time. We'll wait for them, and meantime we're getting ourselves busy in neuro-HIV. But I think there's a lot of information you can get by studying actually peripheral events, and especially what happened in T cells. So without much ado, the crux of the idea is that we succeeded to infect immunocompetent mice. So you can use any mouse strain, and that opens a big field of mouse genetics to this work. Because I'm probably the oldest among you, I give you a tiny historical perspective, and some of you obviously know that HIV pathogenesis, and I think that's my assignment for this talk, how we can apply this model for HIV pathogenesis and not just neuropathogenesis, had two distinct periods, as you know, sort of pre-art and post-art. Right, and they kind of the breakdown is kind of year 2000. And AIDS obviously started in 1982, and there are consequences to this because the disease under art is very different. than it was prior to ART. Of course, initially the hope was that the disease will go away with ART and we can prevent it or cure it, but it did not go this way. And we have now basically persistent of HIV under ART and frequently see multiple actually chronic diseases associated linked to HIV in patients, and that includes nervous system, which is my major interest, digestive, vascular, respiratory, and almost wherever you look, people look at heart, people look at this, they find, at least in our model, they can find some pathological changes. So quite broadly replicates some of these diseases. So the coming back to my track, with ART, the goals of HIV research changed. Now, I think the primary goal, including our governments, is HIV cure and eradication. And at the same time, we want to understand pathogenesis of those chronic diseases, because some of them are nasty, even though cognitive impairment is not dementia. It changes with age. I will have a slide on this. Some of us entertain the idea of functional cure. So I believe it will be very difficult to eradicate HIV completely, despite some fancy methodologies used. And I think it would be at least a relief if we can cure or reverse some of those chronic diseases and just let people live with HIV on long-term ART. And the last one is our old friend HIV vaccine. And you are all aware what happened with COVID, right? The vaccine in half a year. Well, the first vaccine was tested in HIV field in 1992. So we almost have a 40 years anniversary of trying to develop HIV vaccine. So it's extremely challenging, a separate field, NIAID, and I respect my colleagues and let them struggle. But my feeling is that you do something the same way, it doesn't work. Something has to be changed, but I'm not going to advise them on vaccination strategies. So challenges are the shift I indicated from lethal HIV infection, that essentially it was a death warrant, to lifelong HIV persistence under stable ART with very low virus burdens. We argue higher here, lower there. Functional immune systems, normal lifespans, a kind of relatively healthy life, but high frequencies of chronic illnesses that I mentioned. So that's fine, but with this sort of disease pattern or infection pattern, clinical studies on HIV pathogenesis became very difficult on two accounts. One, most of people feel quite okay. They forget where their keys are to the car and so forth, but that's fine. And there's no incentive to participate because it's not a deadly disease anymore in those huge trials when they want to bleed you every 5 minutes or take your CSF samples, right? So you have to recruit a lot of patients to account for genetics and drug abuse and everything. And you have to follow it up for a long time because many of those diseases that do not develop right away. So people go on ART, we know that they can develop cognitive impairment in half a year or a year, and then you want to follow it up. So it's really almost life commitment to sit on, participate in those studies. So as we know, many basic research questions on HIV under these conditions can be addressed in cell or organotypic cultures, but then you have to have, of course, ART, and like in several models of latency, and you still have to confirm it in real life in some cohort. So all that says, hey, let's study it in animals, but that's not easy either. The existing models were difficult to adapt, and I'm not sure how good they are. I'm not propagandist for in a sense I am, for our own model, but it's not easy with the existing ones. What this slide is doing here. Okay. Before I go to the models, I just wanted to go back to my own field just to exemplify to you how the disease changed. As you know, HIV dementia was a big problem prior to ART. That went away almost. It's very low percentage, and there's mild cognitive disease, which we call mild hand. I like to call it NCI because it's really mostly cognitive impairment, and it's really in relatively healthy people that can function and do things, and they sort of have cognitive impairment of low grade initially that may even resemble very, very early stages of Alzheimer's, which is called MCI, right? Mild cognitive impairment. And that field is arguing about timing of that as well. So, but it's relatively early compared to dementia, which was end-stage disease. Severity, as I said, is mild, but increases with aging. And then as people with HIV live longer, I think the average age now is 55 years. They begin to enter the age bracket when a serious age-related neurological neurodegenerative disease develop. And if there is a pathogenic interaction between aging HIV and other diseases, it's a big question now, and we actually are trying to address it with HIV as well. And it's difficult to test these people. It's really a neuropsychological test, which is objective, but takes time, and it's what it is. And brain scans, you now do not have with mild HIV the reference brain changes that were so easy to obtain with dementia people. So, so that's a big problem, and we want to access their CSF, cerebral spinal fluid, but that's an unpleasant procedure. Not everybody agrees. So main manifestation, cognitive impairment, which has a scale. And the mildest one is one standard deviation over normal. Another surprise, brain pathology, minimal or normal. Susan Morgello calls it really normal. Minimal cell activation, inflammation, altered energy lipid metabolism by large-scale methods, some CSF metabolomic changes. Others, and then we mice found that there's minimal neuronal apoptosis, and it's really depending on the comorbidities in aging. But that's all hopeful because the neurons basically don't die en masse, so you can try to cure that disease. For virologists, it's a big problem. Extremely low HIV brain burdens. You know, my lab is 2 floors over Susan Morgello's, and they have a project to quantify HIV in brains from people with unsuppressive ART, and they just can't. They even with RNA scope, they're having difficulties to find virus. So that's it's our local problem, right? And the pathogenesis is not easy to determine. So again, animal models would be good. So coming back to the animal model highway, as we know, infectious macaques and cats with FIV, macaques with SIV has been a fantastic model for study of immunopathogenesis and progression to dementia. Now, that can be kind of stopped with CART or slowed down if applied within really 24 hours of infection. So you cannot really let it go much. So it's almost like prophylaxis. And even then you have a lot of changes. So utility to study mild hand with the conditions I described here is unproven in my mind. Steven Spector is trying, a lot of people, right? But it's definitely the model is now less useful for that. And it is expensive, and there's a reluctance on behalf of NIH really to support now big private studies. Humanized mice, great model. Again, it was designed to study immunopathogenesis. You implant human cells, they infect, they die, and some remain in latent reservoirs if you use CR. So it's a nice model for that. With problems of limited or variable graft survival times, graft versus host disease. I think you as a CFAR have this model probably, and you know the difficulties besides also being expensive. Big surprise was that with the standard humanized model, there's really not much colonization of brain or CNS by macrophages monocytes, which we believe is a driving force in brain disease and perhaps in many other peripheral diseases. So attempts are being made. There's mouse in Nebraska now that is transgenic for certain growth factors, and more monocytes go to the brain. So people are trying. And then there are all the transgenic HIV or HIV gene, single gene expression models, TAT transgenic mice, NEF transgenic mice, which do nicely reproduce elements of brain disease and obviously directly correlated with specific genes, but they don't reflect the dynamics of the disease. And now we're all interested to put it in some sort of a physiological context of organism that develops disease, right? So many things are involved that this is almost necessary. So single protein studies are kind of out of favor right now. As some of you know, we're getting your grants killed, applications killed because of that. Not by me, but I respect every model. But people really want now a comprehensive picture. So when we started, our idea was to forget about transgenic mice, forget about immunodeficient mice. What if we can infect conventional mice, immunocompetent and different mutants, and have fun and reproduce at least elements of HIV infection? The challenge is, as all virologists know, there is a dogma. HIV does not replicate in mouse cells and mice. So we started against this dogma. And as you know, we modified rather than mice, we modified HIV. I will show it in a second. But what started us in our approach was what Sanji also remember. Basically, Stephen Dukers was involved and Mario Kanki previously. I used to work on astrocytes, and there was a big controversy. Can they allow HIV replication? What kind of restriction there is in astrocytes? And we played with pseudotype virus, and we could show in Sanki's work that you get massive infection. There's no restriction once you let the virus out. So the conclusion, or rather advice, is never give up. If you have a good idea, think about it, try different approaches, but not everything looks as it looks, right? If you dig deeper, we know that. And but with respect to mouse cells, then we said, all right, so we can infect mouse cells. Then you go to 3T3 cells, and you cannot infect them with pseudotype virus either. So we discovered by having, actually, I did not work on mice until then, having a colleague in the building that had spare mice and spare cells, that we use primary cells, astrocytes, lymphocytes, and macrophages. We can infect them with pseudotype virus. Not exactly robustly like HIV, etc., but very specifically. So here you have T cells, and here you have macrophages. And no, here it's spleen cells and macrophages. And you can see that infection is dependent on the reverse transcription, so it's a bona fide infection. In 3T3 cells, it doesn't work, and in those cells, it works. And specifically in spleen cells, we get sort of transient infection, but if you look at the production of P24, it continues. So it was a good start. So you get better results when you infect primary mouse cells rather than playing with mouse cell lines. Eventually, we found cell lines that also respond. So it's very, very, quite a lot of work. So based on this, Mary Jane suggested actually to exchange HIV GP120 with murine leukemia virus, GP80. So basically, instead of changing the receptor in animals, we change the receptor on the virus, receptor protein. We left GP41, of course, because of the RRE element, and called it eco-HIV because we use ecotropic MLV, which means it cannot infect people. It's really limited only to rodents. And that was the issue of safety. And it remains the issue of safety, even despite the fact that it does not infect humans. Our biosafety committee considered its infectious agent 3, risk agent 3, that requires BSL2 and a animal BSL2 work. And they are right in the sense that if you puncture yourself, macrophages would digest this virus, and then it, of course, can take residence in those cells and just pump up bad cells. It will not spread, but it will establish some residence, and not everyone wants to walk with tiny amounts of HIV infected cells. So, so that's the construct, and then we just tested it again. Most of our experiments are now in animals, because not only that mouse cells in culture are infected better, but in animals it works better. So there's a lot of it's just easier. You can test the virus in mice in 5 days. You can then continue with your pathogenesis and disease studies and everything else. This is took us a long time. Okay, so we're all friends, but we had enormous resistance, and that's why I'm gray. And because our first paper on this by Mary Jane was published in 2005, and it was extremely difficult to publish anything, because we always got the response, everybody knows that mice are not infected by HIV, cannot be infected. And it required really stamina to persevere. We began to break through skepticism really in your HIV field. I will just have a few slides because that's not the purpose of this, because it's just impossible right now to study mild HIV disease in many other model systems. It's really reflecting very well the changes and allowing us to go forward. So this infection is just kinetics. Mind you, it's very fast, like in people, within 24 hours, right? If someone gets punctured with a needle, with HIV needle stick, they have to take a ART within 24 hours. There's a tiny peak of infection, and then it becomes integrated and steady. So it does not take over the mouse, like HIV takes over the person without ART. And but you can find the virus using eco-HIV with GFP markers in spleen, macrophages, so that's in spleen, macrophages, and it is there. Okay. Long-term kinetics, and we went with in some experiment up to 450 days, mind you. The virus persists, so that's almost like in people for the lifespan of this host, and persists in T cells, as I indicated, mostly integrated state, and persists in macrophages where it's expressed to some extent in a zigzaggy fashion. The persistent in mice is also shown by I have it on the next slide. Here I have the question of viremia that is often asked, because again, people are very focused on the fact that you can detect HIV in blood. But if you, again, think about this in well-suppressed people, you don't detect HIV in blood. You have to do go to more fancy methods. So viremia is transient, and after that, the virus becomes persistent in the very low levels, like in people on ART, and to the point that there's really no immunodeficiency. So the mice remain healthy, have normal CD4/CD8 ratios. I'm reproduce part of the previous slide because we did some comparison with people on ART in terms of total HIV DNA copies. You can see that it's even numerically very similar in mice versus people. Integrated DNA, again, quite similar. And HIV GAG RNA, again, very low, declines in mice and then becomes tiny. And the same in people on ART. Okay, and even when we try to do integrated DNA to total DNA, the ratio seems to be similar. So that still did not carry much sway with people from NIAID, but we are patient. Regarding ART, the curious and I think very useful feature of the system is that you don't need ART. This is the mouse immune system that controls virus. And so it's technically kind of similar to elite controllers that control virus by the immune system, not the ones that have lack receptors or the number of possibilities in non-progressors. But a fraction of elite controllers have immune system control of the virus. And they, of course, don't need ART either. So it's not sensitive to ART. You cannot do anything with ART anymore. But the virus persists for a really long time in a cycles of reactivation and decline. When it's reactivated, the immune system kills the virus-producing cells. So you can see it in this panel when we tested individual mice for the GAG antibody levels over time, over a year and a half. And it's coming in blips, goes away, then there is another reactivation, and that's how it goes. So the question that we then asked was what cells serve as reservoirs for persistent infection. And then again, I'm referring you to that paper by Gu in 2018 with a lot of work. He found that determined positively that these are CD4 positive T cells, chronically infected mice contain largely latent virus that can be induced ex vivo and in vivo later, he showed, by epigenetic modifiers. So it's, again, like in people, and induction, like in people, does not result in clearing the virus. Okay, so that remains mysterious. Okay, another reservoir are, of course, macrophages. Now I'm saying of course because for those of us working on brain, macrophages and microglial cells are important, and we believe they drive disease. In those mice, really, the virus is not only present in macrophages here in peripheral, but it's expressed. So this is an expressed or tiny expressed reservoir. Expression is not huge, but those cells are expressed, they produce viral proteins, they induce macrophages and microglia to production of harmful proteins and products. So an important reservoir that I think will have to be dealt with if we want to both eliminate HIV and mitigate HIV-driven diseases. So I have summaries from time to time, right? So the dynamics of infection resemble people on ART, but you don't need ART. But you can use it if you want to study artifacts on metabolism, etc. And why we don't need it, that's, we believe, is in part, and we're publishing increasingly about this. Mary Jane, of course, is doing a lot of that work. Effective host immune response against virus. So first, we initially found it by an old method of adoptive T cell transfer. We infect mice for 1 month, 6 weeks, when they have already that immunity against itself, and take the T cells from those mice and put them in uninfected mice, and then infect, and then you can show that you can block infection. Okay, so adoptive transfer indicates that T cells are important, or T cell specific immunity. Others, ourselves a little bit, again, managing in the paper, with vaccines, showed it indirect showed it in a different way by immunizing mice with GACPO vaccine a number of years ago, and we showed that they become protected to a great extent, not completely. That's the weakness of the vaccines. To echo HIV challenge, and the number of people use the model to test vaccines as they develop them against HIV. And a good friend of us, Tom Hankey from Oxford, we collaborate on a number of things. He has very, very interesting mosaic GACPO vaccines that we are testing now as well, and has a number of papers. Some people warn us with their papers, also with ECHO, that that if you include Pol in those vaccines, you actually lose some long-term protection. If you, it's also, they found that persistent infection, that lentivirus HIV induces early myeloid suppressor cells. So slowly becoming, in this model, evident what is not permitting perfect or fully efficacious vaccine. Our trick that we are doing now, what we are trying is to do therapeutic vaccination. So the mice already have their own immune responses, and we use vaccine to try to help them to eradicate virus. Did not work yet, but we're working on it. So that's our vaccine research. Another proof that T cells are crucial as our work in atemic nude mice, which don't have T cells at all, right? And they are very, very infectable with HIV. Indicated role that T cells are necessary for that control. However, we also found that innate immune system is critical for control of, not critical, but important for control of HIV infection mice. Here we worked with interferon, type 1 interferon knockout mice. We confirmed that they are knockouts. And you can see the difference between knockout mice and wild type mice in terms of infection. So if you knock out interferon responses, you get better replication. Again, not virus is not crazy, it doesn't kill mice, but the replication is increased. So we now have growing summary of features that compare HIV infected mice to people on suppressive ART. And these have to be people that are on stable suppression, because as you know, there's a huge variability. Some people cannot take the medications, they change, and they go for a while without, and then get spikes of the virus. So it's all complicated. It's very difficult to work with humans. I don't know if you noticed, but they have their mind, and it's the variability in human population is tremendous. But we in this field are trying now to our clinical colleagues to have proof of suppression that is stable, lasting for years, and then you can get at least that variable out of the way. So viremia is really not, you have to, I've listed a number of things, right? Integration, yes, in both systems. So you have similarities at many levels. Okay. And counting on you faster reading than me talking. So here I'm coming back to the idea that the model resembles human elite controllers. And this is a small group, as you know, elite group. And they are not really, they feel so well that they're not tested for many things, but I discovered that they do have cognitive disease. They have a number of diseases. But it doesn't bother them, and it's difficult to get them into clinical trials. So like in people, ART can prevent infection, but has no effect on persistence and the reservoirs that we defined. And as I indicated, you can really do most of your experiments without ART, but study sections will always say, yeah, you have to have ART as well. And so we do some experiment with plus minus ART and use long-term acting ART that you don't have to inject every day. What Howard Gendelman has now, and GSK actually giving to patients, you can inject ART once a month. So it's extremely convenient for experimentation. Now, so all this control, but the flip side of this is very nice for us because mice seem to be a very good model for both HIV pathogenesis and pathogenesis of the number of abnormalities that are found in people in art, from brain disease to gut disease to vascular, etc. I'm ahead of myself. I just listed papers. So I listed them chronologically, and among the first one was Sabita Roy. We all know, right? And she showed that eco-HIV affects gut permeability, and she's into opioids, so she combined that research. So gut disease, okay. Our friend Sanji showed very early on that there is a blood-brain barrier problem in eco-HIV infected mice. That kind of has been followed by a number of other investigators later. For example, gap junction mediated blah, blah, in brain parasites. So that's Michael Toborek is doing really excellent work with eco-HIV and with respect to brain. So, but a friend a colleague in St. Luke's some years ago wanted to study lung disease, and he really has OPCD disease in those animals. Extremely difficult for him to get funded. He got this paper published after maybe 5 years. And so there's skepticism in some circles to use the model, others, and there's not. So we're breaking through this. And you younger people will enjoy it a lot, I think. And just sheer mass of research will prove to the community that it's useful. And then, of course, we do a lot of cognitive impairment and brain work, and that model is extremely useful in this with or without drugs of abuse. So as I indicated, the macrophages are infected. I'm just give you a few slides of mild hand because it's really promoting myself. We had a lot of fun with this. So like in people, eco-HIV does go to the brain. We believe it's going through monocyte macrophage infiltration. And here we compared infection with eco-HIV versus parental MLV, and you can see a huge difference. So MLV can, to my surprise, replicate in the brain as well, but doesn't cause any disease we can measure. So you have the virus DNA copies, okay? And there is a dissemination, as in patients, in the few brains that we have access to from highly suppressed people, there's really no gross pathology. And inflammation is sort of minimal. There's no pathology by tunnel assay, so very quiet. Ben Gelman and Susan Morgero call it normal pathology, which for me is an oxymoron, right? If you have pathology, how can it be normal? But these folks are deep in their own field. They have terms. So great, normal pathology. However, if you inject virus IC intracerebrally, you can study cell tropism, and then it appears that macrophages, microglia, by their markers, can carry eco-HIV specifically. So that's one of the figures we published. So the main target cell in the brain for eco-HIV is macrophage microglia. And interestingly, MLV does not infect those cells. So something I will not be going much into, we now believe that the cell tropism of eco-HIV in mice is driven mostly by the LTR. And we have quite a lot of work emerging to show why, via what mechanism. So, and as you know, in other retroviruses, LTR is very important for cell tropism. So it's not just surface receptor. In this case, this is an ion transporter in mice. So echo could theoretically infect everything, but it really infects CD4 positive T cells, myeloid cells, and here and there astrocyte, here and there some other cells that we don't bother to look at. So tropism is very similar. And that brings us to the cognitive disease that we published a lot. And what is nice about this is that there is a readout in mice for cognitive disease. So there's water maze tests for hippocampal activity, action in memorization and long-term memory. And this is just an example of what we use very often, radial arm water maze. So it's a maze that has 6 arms, and mice have to decide where the platform is. And many in this field like this because it also measures working memory. And as those of you working on neuro HIV know that working memory as well as long-term memory learning are impaired. These are some of the major criteria. So we like to have this readout. So there's a clinical readout for us in those mice, and that's cognitive impairment and memory loss. I had just one slide showing that art as treatment when the disease has been already established has no effect on that disease, and that's obviously a situation in human beings as well. And then we go and we can just do anything in this system. So we like to do transcriptomics and genomics and whatever, and compare it with human brains, and you can see that there is a lot of genes involved in synaptic activity that are demodulated in the system, and you can test gene families and so forth. And I will not be dwelling on this, but some of it is that outcome of this is, and we did not publish it yet, that is dysregulation of memory acquisition in mice seem to be also due to the effect of eco-HIV disturbing epigenetic control of memory. So that's interesting. We got into this, and at very advanced age, I started to learn neuroscience big time, and that's, if you want a big field, that's the field. So we also found, as again we published, and some of you know, that memory impairment correlates with diffuse dendritic dearborization in hippocampus and cortex. And with a colleague from Colombia, we can do LTP in those mice in hippocampal slices, which is technically tough. We have to send the mice to them the same day, they kill them, they take slices, but it's working. So LTP is reduced, which is also a measure of defective memory. So I often even refer to this as memory defect rather than neurocognitive impairment, which is a big term. And sometimes it's nice to be specific. So in part, it's driven by synaptodendritic injury, and I think it's exactly as in people on art with cognitive impairment. Some people refer to this as synaptodendritic disease. Genetic, I indicated that. And the question how HIV dysregulates that with gene and how, and this is an exciting field that we are, number of people are digging into it. The more they dig, the more difficult it is. But we are persevering. But the key issue is that you always want in your animal model to have clinical disease, of course. Then you can study your disease-inducing agent versus the disease, and you can try to cure the disease. So with NCI, cognitive impairment, we have that. Those of you that will focus on, I don't know, lung disease or gut disease will have that readout. But this is a very useful, and I think that's what number of investigators like in the field. So finally, I think my task was also to talk about interventions. And of course, we have several papers and others on the subject. So I just listed several abstracts. It's almost started several years ago. We started with Barbara Slasher and Norm Howie and Justin MacArthur from Hopkins. that they asked me to join the PPG on testing intranasal insulin therapy in people. As you know, intranasal insulin actually helps memory. I actually took some before I started this lecture. So spraying insulin into your nose helps. And they wanted to see whether we can reproduce it in mice, and then of course study the mechanism of it. So indeed it works, and we published this with them, and so this is one of the first interventions when preclinical model went into clinical study that was never completed because they could not, mind you, and this is a problem, attract enough patients, even at Hopkins, to do fully controlled clinical trial of cognitive impairment, plus minus insulin. But with Barbara, and she's essentially in drug development and a wonderful person. So she was into glutamine antagonists as potential therapy for HIV brain diseases, and that worked out very well in eco-HIV model. And there's several papers on it. It works not only in prevention, it works as a treatment. Colleagues from Canada study proliferator peroxisome. PPAR is the best gamma agonist, and they also have effect on reducing or reversing a lot of inflammatory changes with eco-HIV. They did not do clinical readout studies yet, but they're doing it. Mary Jane got into followed up on the innate immune responses, and this is actually exhilarating. You can train the innate immune system, and even 2 or 3 weeks after that, as it's described in this paper, the immune system will prevent or reduce HIV disease in those mice, as well as HIV levels. So innate immune system is something to behold. NIH has now a bunch of programs about this, and it's gaining respect, right? Because we know this innate immune system, yeah, that's transient, 2 minutes, 5 and 2 days, and then CTLs take over. Well, not necessarily. And with John Berman, we have a project using buprenorphine treatment to prevent NCI in mice, and now more recent studies to actually reverse it. Now, bup, as we call it affectionately, in short, is affecting monocyte entry to the brain. So with her, we are finally getting kind of an absolute proof using some of the excellent, or with help of excellent graduate students from Einstein, that monocytes are critical. Monocyte entry is critical for both initiating brain disease, HIV brain disease, and for maintaining brain disease. So that summary I will skip because you had it in the table. But so far so good. We like to study disease, and I'm all over the place, and I'm sorry. A lot of people want to work with me, and I'm not refusing. And as a result, I really like to. I'm happy to teach people to work with eco-HIV, but it requires some time investment. But I think it's a wonderful model for especially brain issues, but also for T-cell reservoirs. So what we're doing now, we are really focused on monocytes vis-a-vis brain disease. We are beginning to identify molecular determinants of impairments by massive, omics right now, to the point that NIDA actually gave us a UO1 grant to use ECHO. Basically, the goal is to determine single-cell transcriptome changes during chronic infection, plus minus opiates. And we are the only mouse program there, in addition to several human brain programs. So this is done, there will be just gigantic amount of information on changes at many levels and single cell level. in brains of those mice, plus minus opiates, which will be available to everyone. And that will lead probably to a lot of follow-up research. Okay, I'm over my time. Therefore, I'm summarizing. This is a picture from the west side of Manhattan when our lab was until 6 years ago. And then Mount Sinai purchased yet another hospital. It became a very large medical center. And they moved us to that black building in main campus, which I enjoy very much. So our lab contributors are many, but of course, Mary Jane is critical, has been critical in developing this model. We have collaborators in Colombia, and that's for a lot of neuroscience, and now working, trying to merge HIV research with Alzheimer's research through animal models for both diseases. Leroy Scherer, not very active anymore, was critical at the early stages in pathology of our system. Susan, of course, everyone knows human tissues, but she also can tell you what happens in mouse brain. So Susan is enormously helpful. And I have really long collaboration now with Barbara Slasher and Department of Neurologist Hopkins on kind of merging their neuroscience and drug development and our model. And actually they have their model now. They have the model in Hopkins as well as part of their either CFAR or one of those core P30 or P50. So I hope I was not too pushy in telling you good things about the model. There's a lot of problems, of course. It's not exactly HIV. Does not take over mice, but it's useful for many aspects of HIV disease. And kind of luckily for us, because macrophages are the main carriers of the virus in mice, it's very suitable to study mild brain disease and actually other chronic diseases that are developing in people on art. So thank you very much. I'm sorry for taking all this time. And if there are any questions, I would like to respond.
If I may ask a question, I don't know, do we have time for that?
Yeah, thank you so much, David. Thank you. Now it's open for discussion. Michael, yeah, you can jump in.
Okay, thank you, David, for this powerful advocacy for eco HIV. I mean, this was wonderful to hear. I just wonder about these early papers that showed that TAT cofactors like PTEF, for example, from mice are not working very well with TAT. And that significantly limits transcription of HIV provirus. But what you showed appears to be that production is similar to what you have with HIV, right? So I wonder, did you actually test those PTEF from mice, specifically, and how do you answer this kind of concern?
So we were concerned about this, or aware of this from the very beginning, right? That was the major question. Now, again, as I indicated at the beginning, there is a big difference between primary cells and tumor cells, and like 3T3 cells. And if you know that much of that research declaring HIV dead in mouse cells was done in NIH 3T3 cells, because everybody kind of likes to work with them. Well, we cannot infect them well either. So that's why we are so keen on doing work mostly in mice, because in mice, somehow it works. Now, we are getting to, from the tropism point of view, to this question now of LTR and transcription factors. So there's work in progress, okay? The fact is that it replicates very well in macrophages and TAT antibodies, and it works. Now, again, I said at the very beginning, it's not HIV replicating or SIV replicating in monkeys, okay? It's right away starts from sort of suppressed situation, and we We know that the suppression is mostly driven by the immune system, and very useful for some studies. There is likely also to some extent at the transcriptional level. So all that has to be yet fully clarified, but the end outcome, the way I look at it practically, right? And we are all practical also. You infect, you get initial peak of infection that is very powerful, then the immune system subdues it, perhaps transcription, not fully compatible transcription factors, and it settles down to essentially chronic infection in macrophages that the virus and virus products are made, and cellular products that are pathogenic are made, and latency in T cells. And that is exactly what you see in people on good ART, or suppressive stable ART. So with combination of maybe some luck and the fact that now HIV moved mostly to suppression state, it's a mostly suppressed virus disease at this stage. The model is becoming very useful. Okay, we are going to clean up the factors, but I'm doing it kind of aside through, we're getting those big programs to drug war abuse, etc., because it's extremely difficult to get funded on basic studies on HIV now, as you probably know. It's HIV funding is declining, and there's a lot of funding for drug abuse and for vaccine, which I don't understand, right? Because they're doing it for 40 years, and some other aspects, and of course, clinical support. But to get basic research grant funded is very difficult. We tried with NEF, as you know, and we will try again. But now we'll try it after sequencing everything you can find in mouse. So that hopefully will overwhelm the reviewers. But because I just would like to have an F grant. And so we're using funding from here to there to study the transcriptional events. It's impossible now to do it full speed based on the funding. Okay, but we will do it. We're in the process of doing it.
Thank you, David. David, we actually HIV transcription in mouse cells, but we didn't use 3. We used epithelial cells, like similar to 293 cells, immortal epithelial cells, and we did see transactivation. It was not like 40-fold, maybe like 10-fold, but it's still there. But I do agree that P2FB from mice, I mean, if you try to model it, the complex just stay, I think, together because of this single mutation in P2FB. So there must be something else which is picking up for the transcription. But my question was, you mentioned the lung disease, right? So can you, so in this mouse model, do you think there is any lung associated pathogenesis or anything like that?
Yeah, that's studied by Robert. What? There you go. Got my name of skip my name of my friend, who is a lung scientist. He's a physician that works on OCPD, basically, and smoking, and obviously those folks also look for mouse models with HIV, and it worked for him. So there's the paper that he published is very detailed, very good biochemistry, and he is going for full funding, trying to get it maybe through 5 cycles. But now he got scored pretty well. So hopefully in the next round in September he will get it. So there are similarities at the biochemical level, and they try, of course, interventions, and they are very happy with this. Again, driven by macrophages, lung macrophages, and they're doing it plus minus smoking. So it's very interesting research. I'm not into it, but it's nice. I think Michael Toborek is hitting the microvascular disease in the brain, as well as brain stem cells, which again are translatable to human situation. And so that's, I think, vascular issues with HIV and eco-HIV will be a big field for people that will get into it. Gut, I don't know, because Sabita does not have much follow-up. So, and in gut, you hit all this, it's a very difficult research in terms of isolating those cells and so forth. So what else? It's heart, right? Heart disease is a problem. I think echo reflects a lot of it. Yeah.
Thank you. Yeah. We just started our first sections, so we try to test eco HIV and this sickle cell mice. And
anything I can help.
we also
the lab is open for you.
You already did. You already put the virus. So thank you so much. So hopefully we'll get some. We have some data ex vivo, but not yet in vivo.
Okay, and whatever you need. We have luciferase. We have mini luciferase marker now, marker protein, and so forth.
Nice.
So whatever you guys need. Okay.
Great. Thanks so much. Any more questions?
I have one. So it's more like a comment. We all want to cure HIV, and it's a wishful thinking that we have is that once the HIV is gone, all the comorbidities will be gone. But we don't know exactly if that is the case. And none of the animal models can really address that besides the other than eco-HIV mouse model. The reason is that a mouse has a very strong anti-HIV immune response. And so just by reactivation, forget about art or anything, just reactivation will be enough to eliminate several of the viral reservoirs in this mice. So there is a chance that all reservoirs will be eliminated from this mice, and then there would be a small window or a window to see how the disease look like.
Yeah, but why do you think? Do you think that the disease will stay after HIV? I think it will start declining, right? Because we find that it,
If it is the legacy effect of HIV that we kind of implicate in the chronic disorders like neuro HIV, then it is likely that it will stay even milder state, but it will be there.
Yeah, well, I think you are right. We did that with insulin when we withdrew. So insulin was improving cognitive impairment and even reversing synaptodendritic changes. And then you withdraw it and everything comes back. So it's enormously, it's actually very impressive. It's enormously powerful pathogen, right? Whether it's TAT or NEF or God knows what, or this other cytokine, which we have to figure out, right? Yeah. So there will be lingering effects because the comeback was very, very rapid. Yeah.
Thank you.
But again, I'm sorry if I sounded as a propagandist for this, but you wanted it. You wanted it. As you know, I'm not running from meeting to meeting to talk about this, but I think it's a useful model for certain aspects and for really committed scientists, because you will have resistance at some levels. Okay. So that's the main thing.
Interesting. David, have you thought about infecting mice which would express human cyclin T1? So this would replicate human transcription. I think this mice was made like a long time ago.
I know they were made, and I think people in Einstein still have them. But the hope was that cyclin T1 plus human CD4, and the mice will be infectable galore, and they were not. Right? So I think Goldstein was working on this, and it seems to me he gave up eventually. So I think cyclin T1 maybe for sure it's important, but maybe there's a additional element that substitutes for it at least partially, because clearly LTR works.
Great. Yeah, thank you.
Right?
Yeah, sure.
Thank you so much.
Yeah, I agree.
Do you have time for a question? This is Italo Mocchetti from Georgetown University. David, very nice presentation.
Thank you.
So the neuropathology that you showed to us in your model, from our point of view, we can reproduce by using the GP120 transgenic mice, exactly the same. Yet I believe that you do not believe that in your model GP has any role in neurotoxicity.
Well, that's maybe a too strong a statement, Italo. And your work is phenomenal. I know your work. I think there are overlapping capacities of this protein. We found it when we started dig deeper into NEF. NEF, if you use, and we engineered NEF into MLV. So we have MLV NEF, MLV TAT. They kind of do the same thing, almost identical. So I think the virus is smart enough not to rely on has 2 or 3 legs for everything.
Sure.
It's a little spider. So there is overlap. But in my humble thinking, I think the envelope proteins are really to allow virus to enter. The secretion and all that, a lot of this will be a factor of overexpression. I think they work with transgenic TAT mice. Now, once they developed the inducible model, they found that you can express it at a lesser level, and then you get different situations. Then you don't have neuronal toxicity. Actually, you have only synaptodendritic changes. And prior to that, NEF was the neurotoxin, right? So everything in excess is a problem. And on top of it, we have overlap with functions for sure. With such a small virus is very smart. And the vaccine folks are learning it for 40 years now. So it's amazing. So I think that's the rationale. And I fully respect people working on GP120, on TAT, and we never been where we are without those works. I simply reflect that the fact that now there's a tendency to try to get a more global physiological picture. And that's possible with Echo HIV. And of course, it's the first criticism against working with TAT or GP120 alone. How does it work in the context of everything? Right. We are even, I will give you even a small unpublished information here that we started. It looks like the TAT is to some extent subjugated to NEF. If you play with this enough, there's a sequential functionality of these proteins, and NEF kind of can overrule TAT. So it's amazing. We can get behavioral impairment with TAT negative virus.
Right.
Okay.
Thank you.
Super duper, but
no, thank you. Absolutely.
I have a quick question, just a short one, because we are running late. But so I was wondering, since you mentioned that the virus persists, I was wondering if the immune system shows signs of exhaustion and immune activation similar to what you would find in the HIV-infected individuals.
Excellent question, and really, I don't know. We did not dig into this. We, of course, Mary Jane is a trained immunologist, and we could look into this. We also have now an excellent postdoc from Wuhan, of all places, that is a viral immunologist. And I think it's an excellent question. If it's possible to do it quickly, so you would test it in what, half a year infected mice, or how long after infection?
Well, first of all, to see if you have signs of exhaustion in T cells. like
Okay.
you will see in the HIV specific T cells, in the eco-HIV specific and non-specific T cells, by markers of immune activation, exhaustion.
Excellent. We can address that. Okay, we will try.
Yeah, that would be fantastic, I think.
Because that would be another thing to really use in this model compared to humans.
Thank you
Okay.
very much. It was an excellent talk.
Thank you.
Very exciting.
Thank you. My pleasure. It's
Okay, so I think
We can move on.
we start moving
Martha,
forward
please.
with the
Yeah.
other presentations.
Okay,